Hello friends, welcome to my channel Sam Biology Class. Friends, today we will discuss about hot start polymerase chain reaction. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Polymerase chain reaction uses DNA polymerase which is slightly active at low temperature. In conventional PCR, all reagents such as DNA template, DNA primers, deoxyribonucleoside triphosphates, divalent ion, buffer solution and the enzyme Taq polymerase are mixed in suitable proportions at room temperature. It is due to the activity of DNA polymerase at room temperature. Primers may form primer dimers or anneals to the DNA non-specifically. This results in non-specific amplifications. Now let us know what is primer dimer. Primer dimers are double-stranded structure which are formed due to the presence of complementary regions within the primer or between two different primers. There are two types of primer dimers. One is self-primer dimer, one is self-primer dimer, and the other is cross primer dimer, and the other is cross primer dimer. In polymerase chain reaction, two different primers are used. They are forward primer and reverse primer. For example, this is a primer which has two complementary regions. Due to the presence of these two complementary regions, the primer can fold upon itself to form a double-stranded structure like this. So these two are the complementary regions between which base pairing occurs. And this is the free 3' end, and this is the 5' end. After formation of such a structure, the 3' end can be extended by the enzyme Taq polymerase in 5' to 3' direction. Such type of dimer is called self-primer dimer. Self-primer dimer. In addition to this, cross-primer dimers are also formed during polymerase chain reaction due to the presence of complementary sequences between two different primers. For example, these are two different primers which have complementary regions at their 3' end. So here these two regions are complementary, so base pairing takes place between them. After base pairing between the complementary regions of two different primers, the enzyme Taq polymerase binds to the free 3' end, then extend these two primers in 5' to 3' direction. This results in the formation of a double-stranded structure which is known as cross-primer dimer. Formation of primer dimer and its subsequent amplification consumes essential reagents. This results in the shortage of primer and other reagents in the reaction mixture. This process of non-specific amplification, which occurs before DNA denaturation, competes as well as interferes with the amplification of target DNA sequence. Non-specific amplification means the amplification of non-target sequences. This process increases the presence of undesired products which reduce the yield and efficiency of conventional PCR. In order to improve the yield and efficiency, another related technique known as hot start polymerase chain reaction is preferred. Hot start polymerase chain reaction, also known as hot start PCR, is a modified form of conventional PCR, is a modified form of conventional PCR in which DNA polymerase activity DNA polymerase activity is inhibited at room temperature thereby preventing non-specific amplification and primer-dimer formation Like conventional PCR, this technique also employs thermostable DNA polymerase such as Taq polymerase. However, it utilizes additional heating and separation methods such as inactivating Taq polymerase or inhibiting the binding of Taq polymerase and late addition of Taq polymerase to improve the yield as well as to provide higher specificity and sensitivity. Hot start PCR is a sophisticated technique in which some of the reagents are kept separate until the reaction mixture is heated to the specific annealing temperature. This reduces the annealing time, which in turn reduces the likelihood of non-specific amplification and also reduces the influence of non-specific primer binding prior to denaturation. PCR typically consists of a series of about 20 to 50 temperature changes known as thermal cycles. Each cycle consists of 3 different temperature dependent stages such as denaturation, annealing and elongation. First stage is DNA denaturation, also known as melting. In this stage, the reaction chamber of the thermal cycler is heated to 94 to 98 degrees centigrade for about 20 to 30 seconds. At such a high temperature, the double-stranded DNA template is denatured or melted due to the breakdown of hydrogen bonds between complementary bases that result in the formation of two single-stranded DNA molecules from each double-stranded DNA template. After denaturation, the reaction temperature is lowered 50 to 65 degrees centigrade. In this stage, the primers which are present in excess in the reaction mixture is annealed to the 3' end of the single-stranded DNA molecule. In polymerase chain reaction, two types of primers are used. One is forward primer and the other one is reverse primer. Forward primer and reverse primer are annealed to the 3' end of the complementary DNA strands. After annealing, next stage is elongation. In this stage, the reaction temperature is adjusted to 75 to 80 degrees centigrade. In this stage, the enzyme Taq DNA polymerase extend the primers in 5' to 3' direction, thereby synthesizing two new DNA strands that are complementary to the template strands. So, in this way, in one cycle, from one double-stranded DNA template, we obtain two DNA copies. If this process continues for several cycles, then a large number of copies of the specific DNA sequence will be obtained. In hot start PCR, non-specific amplification is minimized by completing the reaction mix after denaturation. By contrast, in conventional PCR, all reagents are mixed at room temperature, because of which non-specific amplification as well as primer-dimer formation takes place. But because in hot start PCR, the reaction mix is completed after denaturation, non-specific amplification is minimized. In hot start PCR, the reaction mix is completed after denaturation by using different methods. First one is use of modified Taq polymerase, use of modified Taq polymerase, which is inactive at room temperature. This involves the use of Taq DNA polymerase. This involves the use of Taq DNA polymerase complexed with anti-Taq polymerase antibodies. Complex with anti-Taq polymerase antibodies. In this complex, the antibody binds to the Taq polymerase and inhibit its activity at low temperature. It is only when the reaction mixture is heated to the specific annealing temperature, the antibody degrades and dissociates, and thereby releasing the polymerase, who is then carry out the amplification process. Second method is the use of chemically modified deoxyribonucleoside triphosphates. Chemically modified deoxyribonucleoside triphosphates contain a heat-sensitive protecting group at the 3' terminus. It is only when the reaction chamber is heated to the specific temperature after denaturation, these protecting groups are removed and then the deoxyribonucleotides start interacting with the DNA polymerase, thereby initiating the process of amplification. In this technique, the modified DNA polymerase or the modified deoxyribonucleoside triphosphates become active only when a higher temperature is reached, typically during the first denaturation step of the PCR cycle. Hot start PCR is highly efficient than conventional PCR. This is because it prevents non-specific binding and the amplification of unwanted DNA fragments, thereby producing a more reliable and clearer PCR result. Third one is the physical addition of one or two essential reagents after denaturation. The essential reagents for polymerase chain reaction are DNA template, DNA primers, Taq polymerase, deoxyribonucleoside triphosphates, buffer solution, and the divalent ion. In conventional PCR, all these reagents are mixed at room temperature, because of which non-specific amplification takes place. But in hot start PCR, one of the methods to reduce non-specific amplification is to physically add one of the essential reagents after denaturation. So by adding one of the essential reagents to the reaction mixture after denaturation, we are preventing non-specific amplification, thereby minimizing the presence of undesired products. Friends, this is all about hot start polymerase chain reaction. For more updates, subscribe my YouTube channel, Sam Biology Class.